Hi, welcome to the Bio and AD Tutoring Center videos. Today we're going to be talking about the endomembrane system, what sets it apart, the organelles that are a part of it, and what sets those organelles apart from the other organelles in the cell, and basically the structure and function of all the organelles. So first of all, what is the endomembrane system? The endomembrane system, all the organelles are organelles that are related through direct or indirect contact via vesicles. So they all have phospholipid membranes that can butt off vesicles, and then those vesicles can go attach on to different organelles in it. And so why do we want membrane-bound organelles? Like, what's the purpose? And this can apply to all the organelles, including the chloroplasts and the mitochondria and the nucleus that aren't part of the endomembrane system. And basically the reason we want these is because it helps us overcome the surface area problem. So a lot of the important functions in the cell happen around membranes, and they require membranes. And so if we want to have more functionality, we need to have more membranes. But having more membranes, to increase the surface area, you are going to increase the area on a greater scale. And so by putting these membranes inside, we're overcoming that, and we're not increasing the area with increased surface area. So for example, these two have about the same amount of red. This one has a lot more area. This one has less area, but just as much membrane, which is what we want. And then the other reason is specialization. It allows them to have different and specialized functions that we're going to see today. So the first organelle we're going to talk about is the Golgi apparatus. So I have this picture of a cell that I drew. It's not amazing. I don't have amazing art skills or anything, but the red is the Golgi. So that's where we're going to start. So the Golgi, its structure is it's made up of membranous flattened sacs that we call cisterna. And the Golgi has two sides, the cis and the trans, and the cis is the side that's closest to the ER, so all this black stuff is the ER. So this would be the cis side, and this would be the trans side. And the cis side is the side that's receiving vesicles, and the trans side is the side that's shipping these vesicles off with cellular products. And then the major functions of the Golgi are just to sort, modify, and ship cellular products. So they're kind of like the post office of the cellular world. And then they also can be used to synthesize macromolecules, such as polysaccharides. Next is the lysosomes. Lysosomes are actually only found in animal cells, so this only applies to like our cells. They are a sac of hydrolytic enzymes. And so because they have all these hydrolytic enzymes, their job is to break down macromolecules into their simpler components and then release them to the cell so they can be used for energy or whatever the cell needs to use them for. And an important thing to note is that the enzymes that are contained in the lysosome, their optimal pH, which you will learn more about, but the pH which they like to be in the most, where they can function the best, it has to be acidic. So in the inside of a lysosome, it's a very low pH of about 5. And so it's important that these are surrounded by that membrane, because if that membrane would break, all these hydrolytic enzymes would get out and eat up the cell, which we wouldn't want, and then the pH could affect it as well. And then just an interesting note is that the lysosomes are created by the rough ER and Golgi. So the lysosomes were in blue in this image. And then the next is the endoplasmic reticulum. So the endoplasmic reticulum is just a bunch of sacs and tubules that are all connected together, and they're called cisterna as well. And the inside of it is called the lumen. We'll get more into that in a little bit. So there's two types, smooth and rough. The only difference structurally really is that the smooth ER have ribosomes on the outside of them. Sorry, smooth ER don't have ribosomes, that's why they're smooth, appear smooth, while rough do. So all of the rough ER is right here, and that's why there's all the dots, whereas the smooth ER is over here and there is no dots, the dots representing ribosomes. Even though they don't have very much structural difference, they have a ton of different functions. So the smooth ER is responsible for the synthesis of lipids. So an example of a lipid is a hormone. So the smooth ER synthesizes sex hormones. So cells that are responsible for lots of sex hormones will have lots of smooth ER. They also detoxify chemicals. So that's why liver cells have a lot of smooth ER, so that they can detoxify a lot of chemicals. And then the last function that we all talk about is calcium storage. They just store calcium ions And you'll learn later about processes that gets the calcium out of the smoothie R that lets it out to perform other functions, and one of those can actually help in muscle contraction. But you'll go more into how the smoothie R stores calcium later. And then the rough ER, because the ribosomes are attached to it, a lot of times the proteins will be synthesized right into the rough ER, and then when they're in there, they'll be modified and folded, because remember, protein folding is very important. Polypeptide is not a protein until it's been folded properly. And some of the modifications they can do is they can add oligosaccharide tags, and that will create glycoproteins, which are just proteins that have carbohydrates off of them, and then they can be shipped to the membrane and be used as membrane proteins or glycoproteins for cell-to-cell recognition. So we were kind of talking about how all of these interact through vesicles, and so a vesicle can butt off of one of these and go exchange cellular products with another, or these vesicles can go attach to the plasma membrane on the outside. And so sometimes you might be asked questions. So the inside of the cell is the cytoplasmic region, and this is the extracellular region. And then the inside of here is called the luminal side. And so what will happen is if there's something in the luminal side and it attaches with the cellular membrane, then it will go on the extracellular side, because it will attach and bud and break open. And so that's how you can have, transport particles out of the cell. And then if you also have proteins or something that are attached onto this vesicle, then when they fuse together to the plasma membrane, that's how you get some of these membrane proteins. And then the last organelle is the vacuole. Vacuoles are kind of just like bigger vesicles that have more functions. They're just not carrying stuff around, but they could be storing stuff or transporting. Plants actually have what they call a large central vacuole, and the large central vacuole has lots of functions as well for storage. It stores pigments, which are unique to plants, and then it also has those hydrolytic actions, so it can break down chemical cellular products like macromolecules, because it doesn't have the lysosome, so it has to have some way to break down those big macromolecules as well. And so large central vacuoles can occupy up to 80% of the volume of a plant. So the last thing we need to cover really fast is the difference between a plant and an animal cell. They're both eukaryotic cells, but they do have some significant differences, and it's mostly in the organelles they contain. So I have a drawing of a plant and an animal cell. They're not amazing, but here they are. And so plant cells contain a cell wall that surrounds the plasma membrane and just adds extra support. And they also have a central vacuole, which we were just talking about, and chloroplasts, which are really important for, they're the site where photosynthesis happens. And only plants cells, animal cells don't do that. We don't have photosynthetic abilities. So plants have all of those organelles, animal cells don't. But animal cells have lysosomes, centrosomes, and a flagella, whereas plants don't. So just make sure you know those differences, and make sure you understand the functions and the structure of all of these organelles. That is all for today. I hope it was helpful and you have a better understanding of the endomembrane system. Thank you.